So let's talk about a few chemical mutagens. So these are chemicals that will cause mutations. Nitrous acid will cause adenine to bind with cytosine instead of thymine. So it messes up base pairing. And the reason for this is because here's our nitrous acid. And when it interacts with our adenosine nucleoside, what's going to happen is it changes the chemical group up here. And with a different chemical structure, adenine is going to bind with cytosine instead of thymine. So the structure is changed, therefore its binding relationship and its function is different. So here you have your non-mutated normal parent DNA that is exposed to nitrous acid. It alters the chemical structure of that adenine. And after replication, one of those daughter strands is going to have incorrect base pairing. So where the adenine was, instead of a thymine, there is a cytosine. Now the other daughter strand made from the template is fine because it used the thymine as a template instead of the adenine. Remember, because DNA replication is semi-conservative, whenever you have a new DNA strand, half of that DNA came from the parent. Like it's the same physical nucleotides as the parent. But this mutated daughter DNA strand will end up getting replicated as the cells go through their mitosis or binary fission, and that mutation will continue. So because this adenine is mismatching, you'll get another mismatch with A and C. And then we count this strand also as mutated because this cytosine should never have been there. It should have been a thymine. It should have been a T. So this GC pairing in this daughter strand or the granddaughter strand it's a mutation. It should not be there. So nitrous acid actually changed the chemical structure of a correct nucleotide into an incorrect nucleotide. So it altered our own nucleotides. Nucleoside analogs are chemicals that are so similarly shaped to our own nucleotides that the machinery, our DNA polymerase and the associated enzymes, mistake that chemical, that nucleoside analog, for one of our own DNTPs. For example, we have our adenine, and then you have the analog, the 2-aminopurine nucleoside. And it's just a little different, but it's going to incorrectly base pair. Because this functional group is different, the adenine is going to base pair with cytosine. So you're going to have a AC pairing, and then in the downstream granddaughter strands, you're going to have a GC pair where you should have an AT pair. Here's another example. This is an analog for thymine. So it is our 5-bromouracil nucleoside. So you have a substitution where there's a bromine put into the chemical structure, and then the thymine will mistakenly base pair with cytosine. So again, you get a CG pair in your downstream DNA where you should have an AT. 5-bromouracil nucleoside is actually useful in cancer treatment because you're actually trying to kill the cancer cells. So the cancer cells will incorporate this nucleoside analog instead of the correct thymine nucleoside. So now moving away from chemicals, but still in the realm of mutagens, we're talking about radiation. There's two groups of radiation. There's ionizing radiation and non-ionizing radiation. Ionizing radiation includes X-rays and gamma rays. And these rays will oxidize our nucleotides and cause the breaking of our DNA backbone. So they can cause double-strand breaks, which are really bad. And then in our non-ionizing radiation, this includes UV radiation. And we spend a good amount of time talking about UV radiation. When UV light hits thymines that are right next to each other, adjacent thymines. They cause them to form covalent bonds with each other. And that's called a thymine dimer. And thymine dimers cause problems for the machinery that we use to read DNA. So they will cause mutations if not dealt with. Thymine dimers cause mutations. But a lot of living things are exposed to UV radiation from the sun. So it's not surprising that there are systems that have evolved to deal with these thymine dimers and remove them from the DNA so that mutations don't build up and kill off the organism. We'll talk about two ways in which you can fix thymine dimers. The first one is using photoreactivation, and this is a prokaryote specific system. We cannot do this. Humans cannot do this, unfortunately. It's pretty cool. So photoreactivation uses a special enzyme called photolyase. So photo for light, and then lyase, that's the enzyme that will break bonds without hydrolysis or ATP. So once photolyase has separated the thymine dimers, the DNA sequence is fixed. They are no longer in a dimer, and they are correctly pairing with the adenine that is across from them. The energy that powers the breaking of this bond is actually visible light. So not UV light, but within the visible light spectrum. The organisms that are able to do this can use that visible light to repair those thymine dimers. In our UV lab, we might investigate this. Sometimes we do and sometimes we don't. In our nucleotide excision repair, there is no photolyase available. Instead, the thymine dimer is going to get cut out of the DNA, and then correct thymines are going to be put in place. So we're going to use endonucleases because we need to cut within a DNA strand. So cut the backbone on either side of the thymine dimer. Then you can remove that and it will get degraded. Then DNA polymerase will come and bind to the double-stranded DNA in front of it. So no need for an RNA primer. It can bind to the double-stranded DNA and then fill in that gap. Now remember that DNA polymerase doesn't join the backbones with the phosphate bond of adjacent DNA molecules like this. So you will need DNA ligase to come and connect those nucleotides at the backbone with the phosphate bond.